Hello. Can you guys hear me and can you guys see the screen? Can someone confirm? Okay, thank you. Okay, so we'll start today. Uh, Today we have session 5 where we'll cover lectures 13 to 15. So yeah, important links as always. So in today's class, we'll discuss population ecology. We'll discuss these three lectures: population parameters and demographic techniques, population growth and regulation, and population studies and its applications. And the next module, we'll cover community ecology. So we'll start with week 5, that is population ecology. So the first lecture was on population parameters and demographic techniques. So first we'll start with the basics. What is a population? All animals of the same group of species, same group of species which live in a particular geographical area and have the capability of interbreeding is termed as a population. We have been looking at this definition for quite some time, and I think all of you are familiar with this by now. So we have populations. So for example, we have population of humans, we have population of animals like deer, tiger, and sambar, cheetal, and all that, monkeys. So basically, sometimes just a second. Sorry, show the pen. Yeah. Okay. So yeah. So many times in nature, we want to manage populations. So for example, uh, India is actively trying to manage the number of tigers that are found across the different areas in India. So to manage the population of tigers, we also need to manage the population that is deer, cheetah, and sambar. So many times we might have to manage multiple populations of animals. And to manage a population, we first need to understand most basic equation that you can have of mm, a population, or the most basic model that you can have of how a population changes. So here basically we have Pn plus 1, which is the number of individuals at time n plus 1 or generation n plus 1. Number of individuals in generation n, which is Pn, plus the natality plus immigration minus mortality minus immigration. Basically, let's say we have a population in the n generation Pn. So there are four major forces that can act on it. One is basically natality, which is basically birth or the birth rate per 1,000 organisms are born. And the second thing that increases this number of individuals is, so immigration is basically when organisms from another geographical area move into the population under study. So for example, it's cheetal in this region. And in some year, we have another region from where a lot of deer migrate here. So basically, what is happening? Some X number of individuals of deers are moving from this geographical area to this geographical area. So termed as immigration. whereas the efflux or the removal of organisms to this geographical area is termed as emigration. So emigration is basically individuals leaving the population or moving to somewhere else. And mortality is basically mortality is basically number of deaths per year. So the death rate of the population, like how many individuals are dying per year per 1,000 individuals. So this is a basic equation that governs most of population dynamics. The other thing that we need to understand is something called the PDCA cycle or the Deming cycle, which basically tells us how we can manage or plan our programs to like manage populations and other things. So basically, we have these 4 steps that we do. The first thing is plan, and planning basically involves what do you say? Uh, Basically assessing what is the number of individuals of a given population, if it is high, if it is low, deciding whether it is high or low, or things like that. So that is known as planning. Then doing is basically after we have planned, we have thought of something, we formulate a strategy to, let's say the population of deer is falling in a place, but we want to increase the population. So we plan something to increase the population, such as uh, let's say we increase the amount of resources or we plant artificial grasses or something in that area. So the next step is doing. And after you do or implement something, you need to check or evaluate how well your intervention is performing. So for example, let's say the same example, we have an area where there are few deers, but we want to increase the number of deers. So what we do is we plant more grasses and herbs that these deer can eat. So we plant it for, let's say, 2 or 3 years. And after, let's say, 2, 3 years, we again check how many deers are there in the area, if it has increased or if it has decreased. So that is basically checking or evaluating. And based on that, if we are getting an appropriate response, we'll continue with our plan. Or if we see that even after putting grass, the number of deer population is kind of decreasing or remaining the same and not increasing, then we might think about doing some other interventions or following some other methods. So this is basically the PDCA cycle. And the important thing is that in all the steps of the cycle, if you uh, want to basically take any decision, any informed decision about what you are doing, what you should do, and how it is affecting the population, you need to know the numbers of individuals in a given population in each of these steps. So that is the main point of this. So to follow the PDCA cycle, we need numbers or the population estimates or the number of individuals of organisms at each step when we are managing a population. So yeah, basically, yeah. If we have, we need in the planning stage, we need numbers of individual or the population sizes so that we can decide if we need interventions or if we don't need any interventions, then when management interventions are deployed, we need to check the efficacy or evaluate how they are doing. So that is a check step where basically we are again checking the number of wildlife or animals that are present, which will further help us optimize our intervention. So this is the second part. The third part, which is the act, and basically the fourth part, which is act. So basically you plan something, you do it, you check if it's working, and then in act, you're basically modifying it to make it better. Right. So yeah, as I was telling, numbers and assessment of numbers is quite crucial for everything. So as mentioned before, depending on the existing number of individuals in a population, we might want to increase the numbers if the numbers are getting low, or we want to reduce the number if the numbers are getting high, or if we want to maintain the status quo if the numbers that are there are adequate. Right. So till we know exactly what the numbers are, we cannot basically take any action. So that's why we need the numbers. Uh, so Another important reason why we need numbers is to predict if there is a risk of a population crashing or a population declining very quickly or like going extinct. When the number of individuals in a given population is very low, the population might crash due to various reasons. Some of them are demographic stochasticity. So demography is basically like population structure and how it changes. So demographic stochasticity is basically variation in parameters that affect the demography of the population, or the basically how many individuals are there and how they are distributed. So if we have a very low number of populations, then chance variations in the number of births or deaths might lead to there being more number of deaths than more number of uh, births, and that sometimes leads to basically population crash. And the other thing is, if there is a very biased or skewed sex ratio in a population, and the number of individuals are very low, that also leads to a population crash, because we don't have enough organisms enough other organisms of the same species that the animals can mate with, right? So these are known as demographic stochasticity. The second thing is environmental stochasticity, or random things happening in the environment, such as there might be droughts, or there might be a flood, or there might be a famine, and like these animals don't get any resources, or they're drowning, or there are some diseases or parasite infestation, which is basically leading to a decline in the population. And if the number of individuals is very low, the population might crash due to these things. The third thing is genetic problems. So this consists of three things. So the first thing is inbreeding depression. So generally, when you are breeding individuals that are genetically similar, a lot of harmful recessive genes tend to get amplified. So what happens is these inbred populations start developing diseases and other genetic defects. One very famous example of this is hemophilia in the royal family of England. So many of the descendants, or many of the uh, people in the royal family have hemophilia, which is basically their blood does not clot easily. And that is something that has been attributed to inbreeding depression, because these royal people tend to marry within their royal families and stuff like that. Yeah. The second genetic problem that might arise is something called genetic drift. So, for example, in a very small population, let's say you have a population of beetles, which has where the individuals of the beetles are very low. So, for example, let's say you have 10 beetles, uh, three of them are, let's say, this triangle denotes red color, three of them are red, and actually three of them are triangle, and let's say five of them are square, right? So randomly, it might just happen that these individuals might not get a chance to reproduce because there are not enough of them, or there might be some other random effect happening which probably kills them, or like a few of these, and like this variation is wiped out from the population. So genetic drift generally leads to basically population decline and decrease in variation of the population. And the third thing is loss of heterozygosity. So generally, when you have small populations, they tend to become homozygous, and a lot of recessive genes manifest, as I was telling before, leading to basically inbreeding depression also. Other kind of effects that might also happen to populations that are very low in number are behavioral problems and the alley effect. So the alley effect is basically so generally, in the competitive viewpoint of animals, it is said that if the population of animals is quite high, then due to competition between individuals of the same species, the numbers might decline. So less number of individuals might lead to more growth in the population than very high number of individuals. But the scientist Ali also pointed out that if the number of individuals is very low, then some functions that these individuals carry out as a species or as a group might not be happening properly, thus leading to decline in their population. So, for example, we can take that in case of let's say, pack hunting animals such as like wolves. When individuals, so let's say a wolf group has, let's say, old, there are like two populations of wolves. One has, let's say, around 50 individuals, and let's say it has uh, like 50 adults, and let's say 10 pups or young ones. Right. And we have another population where there are just 10 adults and basically six pups. Right. So in the first case, what happens? These 50 individuals can afford to basically leave 10 individuals to take care of the pups, and then 40 rest of the individuals will go out hunting and will basically catch prey for all 60 of them. Okay. And since this is a large, big group, they are able to catch like bigger predators and, sorry, bigger prey and get like more amount of meat. And they're also able to hunt more efficiently because there are more individuals involved in the hunting process. In this case, however, since there are only 10 adults, so all of them need to go for hunting. Otherwise, the amount of meat that they might or food that they might gather or collect might not be enough for all 16 individuals of the population, right? So all of them, let's say, go for hunting, and the six pups are then left alone at the home or at the cave or something, where basically they have to fend for each other, fend for themselves, and this might lead to like predation of the pups and uh, lower survivability of the pups, and even like if these 10 adults are not able to forage efficiently or hunt efficiently and get enough meat, that also might lead to development of diseases or basically deficiencies and lead to decline in the population size. Right. So if there are very small number of individuals, they are also behaviorally affected. The other behavioral effect might be when like individuals are very low in number and very highly dispersed. So like single individuals are very far apart from each other. So some of them might not be able to find mate or reproduce. So that also leads to a decline in the population number. Sorry. So once we have the numbers of individuals in our population, then we can basically try to plan scenarios and plan methods to basically maintain the number of individuals or increase or decrease the number of individuals. So, for example, let's say we have a population of cattle. So cattle is a very important farm animal. So let's say we have a population of cattle in a village. So there are a number of different cattle, and they are currently going through, let's say, they're having a lot some disease epidemic or something, right? So a lot of these cattle are getting sick and dying. So the we need to plan something and like basically take steps to prevent the population from crashing so that we have enough cattle to get milk and for our own uses. So there are two kinds of steps that we can take. One is called as adaptation. So basically adaptation is when the population is made supple enough so that it can respond to the changes. So for example, in this case, we have uh, cows or cattle, and we have the problem that they are diseased, right? So one thing that we can do is that we can put something like a vaccine. So we can get these cattle vaccinated. So that's one way in which like we are modifying the population and making the individuals like supple enough so that they can handle the disease. So even if there is a disease, they might not get, so there is a less chance that they will get it because they are vaccinated, right? And the other thing is the other kind of step that you can take is mitigation. Mitigation is basically when you are analyzing the causes of why these things are happening, and they are addressing the cause instead of making changes to the population. So, for example, let's say we realize that this disease is caused by a parasite that is staying outside of the by ectoparasite that is basically getting attached to the cows. So, one thing that we can do is basically go around and pick out these parasites and like kill them. Right. So that is a step towards mitigation because that addresses the causes of the thing that is happening, such as the disease. Whereas in adaptation, we are changing the population so it can adapt to the disease and like survive better even if the disease is present. So there are two things that we can calculate for populations. One thing is basically the numbers, as I was saying. So, for example, we can estimate in India what is the exact number of tigers or approximate the number of tigers that are present, or like every year. So, let's say in 2006, what is the number of tigers? In 2010, what was the number of tigers? 2014, what was the number of tigers? And we are basically monitoring it across years. So that is one way to monitor population. The other thing we can look at is basically trends. So in this case, basically numbers is basically, let's say there are 1,000 tigers in 2006, there are 1,014 in 2010, there are 1,060 in 2040. In 2018, let's say there are 980. So trends are basically uh, not for a particular year, but we are comparing between years or different time points, and we are trying to say how is the population changing between these two time points. So basically, trends we can have between, let's say, 2006 and 2010, the population is increasing. So that is a trend. And if we compare 2014 to 2018, the population is decreasing. So that will be a trend that the number is in general increasing or the number is in general decreasing. Okay. So do we need numbers or trends to manage populations? So we need both actually. Sometimes we need, sometimes trends are more helpful and sometimes numbers are more helpful. So trends are more helpful when we need to basically analyze a very large population and we don't exactly know want to know exactly what the population numbers are, but we want to know if the population is increasing or decreasing. So, for example, let's say we go to a forest and quantify the number of sambas that are there. So, the number of sambas, let's say, might be 10,000, or it might be 10,020, right? So this exact number does not matter to us that much because it's a very large population and whatever numbers we are estimating are, of course, like approximate values. What we need to see is across years, is this number of sambas that are there increasing or is it decreasing? Because that is the thing that will matter to us. So even if it's a increase of 20, it's an increase. So the trend we are seeing is it increasing or is it decreasing? We are not concerned with exact how many numbers that are. On the other hand, when there are populations of animals with like very few individuals, so populations which have reached their critical limits, or when an animal is like a priority for us, priority species or a species that we want that's very endemic or only found in that region, and we want to conserve that species. In that case, we would uh, generally do a proper census to get the exact numbers. So, for example, for tigers and for Great Indian bustards, which are like priority species of animals that India is trying to conserve, we generally have census and we try to estimate the exact numbers of these individuals and where they're found. Right. Another thing to note is that numbers are finer data than trends. So, for example, let's say in this case, we have the numbers. We can use the numbers to build the trends. So here there's 1,000, here is 1,014. So we see that it's increasing. Here again, 1,014 to 1,060, we see it's increasing. 1,060 to 980, we see it's decreasing. So from numbers, we can get at trends, but we cannot use trends to go back into numbers. So numbers are more finer data and trends are more coarse data. And we can use numbers to get at trends, but we cannot do the vice versa. Right. Does anyone have any doubts till now or questions? Okay. So the next thing is, uh, so we want to estimate the size of the population and other demographic factors. So what demographic information are we exactly trying to get? So we are interested in many things. One of them is basically population size, which is the number of individuals that are present in a given population. The other thing is basically population density, which is the number of individuals in the population per unit area. So, for example, let's say we have a patch of forest and we are estimating how many, let's say this forest is around 500 square kilometers. So we first, to get the population size, we see how many tigers are present in this forest, right? So let's say we estimate that there are 50 tigers in this patch of land. So 50 is the absolute population size, right? And to get at the population density, what we do is 50 divided by 500. So number of individuals per unit area. So this will give us basically 1 by 10 is equal to 0.1. Yeah, 0.1 individuals per square kilometer. Or since this does not make a lot of sense, we can't have like fractional individuals. What we can do is we can basically multiply it by 100. So we have 10 individuals per 100 square kilometers. And this is now known as the population density. It basically tells us how packed a given geographical area is with the given species or population. So here are examples of some common population densities. So starting from diatoms, which are like um, small microscopic creatures, to like soil arthropods, trees, deer, and people, we see that the density kind of tends to decrease as the size kind of tends to increase, and the density of population of different species can vary quite a lot. So here we have somewhere around 5 million numbers of diatoms per cubic meters, whereas here, let's say when we are comparing people, we have only like 0.03 people per square kilometer in a place like Greenland. Right. So this shows that how varying these population density might be for different species. So population density again can be of two types. So absolute density is basically the exact number of individuals per unit area, whereas relative density is like again like trends we are talking about. So let's say we are comparing two areas, area A and area B. Area A, let's say, has X organisms, like X tigers, and area B, let's say, has Y tigers. Okay. So figuring out if X is greater than Y or Y is greater than X is basically the relative density. So we want to know if area A has more tigers than area B or area B has more tigers than area A. We don't want to know the exact numbers, but we just want to know like if the density of the population is higher here or higher there. That is known as relative density. So how do we measure these like population density? So one way to measure population density is basically total count. So for example, we have census that is carried out in India every 10 years. So in census, what happens is the government sends officials to every house, every place. They go and they basically inquire people and basically count the number of people that are present in each house in each given region and all that. So we have a total count of all the people that are present in India. So that is basically a census. The other thing that we can do, census is basically like counting all the individuals of a population. The other thing that we can do is known as sampling. So sampling is basically when we count the number of species. So let's say there's a huge area, there might be a lot of deers. So in sampling, what we are doing is basically we are focusing on a smaller area or multiple smaller areas, and we are estimating the number of individuals that are present in these small areas. And basically, we are trying to extrapolate that to the whole population. So we are not exactly calculating all the individuals of the population, but we are counting a subset and we are extrapolating it to the whole population to get at the number of individuals or the density. Right. So sampling can be done by quadrats or capture recapture methods. So we'll talk about them in detail now. So we'll first talk about like quadrats. So yeah. Uh, Basically, the objective of sampling is that to get a very representative sample of the population and to reproduce important characteristics of the population as closely as possible so that we can estimate the number of individuals or the population density as accurately as possible. So, for example, let's say we have area which is very homogeneous. So, individuals are distributed throughout. Right. And we have another area where basically we have in this part of area, there's a high density of individuals. Whereas in this part of area, there's a very low density of individuals. Right. So quadrat sampling is basically you divide this area into very small segments or plots called quadrats. And you basically lay out a few quadrats here and there. You basically, in each of the quadrats, you estimate what is the number of individuals that are present. So, for example, let's say in these three quadrats, since this is a more homogeneous area, in these three quadrats, we get similar values. Let's say we get 10 individuals in these quadrats. And we know that this area is, let's say, 1 square kilometer. And the area of the whole place is, let's say, 100 square kilometer. So now what we can do is we are finding 10 individuals in 1 square kilometer. So we can estimate how many we'll find in 100. We'll just multiply it by 100. Right. So 10 is basically the average of all these quadrats. If we do the same thing here, if we randomly place quadrats, okay, and we basically calculate the number of individuals and we try to estimate. So if we randomly are placing quadrats and we are placing all of them here, then the estimate that we will get will be very low because there's a place here which has a very high density of individuals, which is not getting calculated or factored in because we are not sampling there. On the other hand, if we sample more locations like this, we might get a higher value for uh, population density or population size. And that is also something we don't want. So ideally, the objective of sampling is to secure a sample which will represent the population and reproduce important characteristics. So for example, let's say this area is 40% of the whole area is densely populated and 60% of the whole area is not so densely populated. So in this case, we can put random samples in this ratio of 2:3. So basically we have two quadrats here and three quadrats here, and we are estimating the population density using these five things. So in that case, we'll get a more representative sample because our samples are also distributed in a way that the population characteristics are also in a way that the population is also distributed. So the sample is reproducing the important characteristics that are present in the population. Right. So when we are sampling, we have certain things called, for example, things like a sampling unit and a frame. So a sampling unit is basically mm, it can be like administrative units or natural units or like topographical or geographical sections or subcompartments of the area we are trying to basically sample. it can also be plots of definite shape and size. Uh, yeah. So basically, a whole area is divided into small of these units called sampling units for the purpose of sampling. So, for example, if you have this big area here of forest that we want to sample, what we can do is basically one way we can go about it is divide it like this, like grid it. So we have now 3, 4, 5, 6, 7, 8 sections. And these are basically our sampling units. So all 8 of them are our sampling units because we have divided the area into small units so that we can sample those units. Right. List of all the sampling units that are present in our area. So for example, here the list 1, 2, 3, 4, 5, 6, 7, 8 is basically called our sampling frame. List of all the sampling units is called a sampling frame. Now, what is a sample? A sample is basically one or more sampling units that is selected from the population and where we basically go and measure the density of the animals. So, for example, again, if in the previous case, let's say we have 1, 2, 3, 4, 5, 6, 7, 8, and let's say randomly we choose that we'll sample 2 and 6 and 4. Right. So now these three, 2, 4, and 6, will be called our sample. And we are basically getting the population density or measuring the population density in only these three places. Right. The intensity of sampling is defined as the ratio of the number of units in the sample to the number of units in the population. So this whole thing will be our population. These three will be our samples. So we have 3 units in our sample versus 8 units in our population. So the sampling intensity in this case is 3/8. So a higher sampling intensity means that we are sampling more of the population, and a lower sampling intensity means that we are sampling less parts of the population. Again, for sampling, there are various different kinds of sampling that you can do. So such as simple random sampling, systematic sampling, stratified sampling, multi-stage sampling, and PPS sampling. We'll look at them. So simple random sampling is basically where we are choosing sampling units, picking sampling units randomly, so that each sampling unit uh, has the same chance of being selected. So, for example, let's say in this area, we again have these small sampling units, and we are basically randomly. So, let's say we number all this 1, 2, 3, 4 till whatever is there, and then we basically make chits out of it and randomly pick out those chits. And let's say we get 3, 6, 3, let's say we get 12, and we get 14, and we get 5. So we made 19 chits. Out of that, we picked 4 chits. And these are the 4 chits that we get. So we'll sample in these 4 places. So that is known as random sampling. So since we are as likely to pick any chit, uh, so like the probability of picking one chit is the same as the probability of picking another particular chit. So that's why this is called random sampling. And basically all the sampling units have the same chance of being picked as a sample. In systematic sampling, what we are doing is we have a simple rule that we'll select every kth unit starting with a random number chosen between 1 and k. So for example, let's say we have 20 sampling units, 1, 2, 3, 4 till 20. And we decide that our k will be, let's say, 5. And for the initial uh, thing, we have to choose a random number between 1 to k. So 1 to k, let's say we choose 3. So what we'll do here is we'll choose 3, then 3, 4, 5, 6, 7, 8. So we'll choose 8. 9, 10, 11, 12, 13, 13, 14, 15, 16, 14, 15, 16, 17, 18. So these are the 4 samples that we'll pick out of 20. So basically, we are systematically picking samples instead of randomly picking them. But the starting sample that we pick, the first sample that we pick is still random. Okay. Now, stratified sampling is, as I was telling you before. So let's say we have a area where 40% of this area has a higher population density and 60% has a lower population density. So in stratified sampling, we make sure that 40% of our samples are coming from here and 60% of our samples are coming from here. So we'll have 3 sampling units here and 2 sampling units here to maintain that ratio. Okay. Multi-stage sampling is basically first we select very large units or very large sampling units and samples, and in between in each of these sampling units we make more smaller samples and then we select those. So, for example, if the area we are trying to estimate the population of animals is quite large, then what we can do is we can broadly divide the area first like this into, let's say, 8 segments. Then each of these segments we can divide into, let's say, we randomly pick the second segment and the 7th segment. Then we'll basically grid these segments and randomly pick out small sampling units. So this is known as multi-stage sampling or subsampling. PPS sampling or probability proportional to size sampling is basically used when the units are varying in their size. So, for example, let's say we have trees. Let's say we have a small tree, we have a medium tree, and we have a very old, very large tree, right? And we are trying to, let's say, estimate what is the biomass of this population or something like that. So, what is the um, population density or biomass? Let's say we are trying to estimate. In this case, what we need is like we might miss out a few individuals of these smaller class. So we might miss out a few of them, and that is okay. But we cannot miss out any of these larger individuals because they contain a large amount of biomass. So the thing that we are measuring in this case, which is basically biomass, is also proportional to the size of the organism, right? And the organisms are varying in their size. So here, what we want to do is we want to basically weight picking of the individuals based on their size. So we'll basically pick out more individuals of this size compared to this and this, or some other thing based on what exactly we are trying to get at. Right. So this kind of sampling is known as PPS sampling or probability proportional to size sampling. Does anyone have any questions regarding these sampling methods? Okay. So the next method to measure like population density or population size is something called the capture recapture method or the mark capture method. So basically what we do is, let's say this is the whole area. We go to a particular place and we basically capture some individuals. So let's say we are looking at lizard species abundance or like what is the population size of let's say some rock agama or some lizard species. Then what we'll do is in this area we'll try to capture as many lizards as possible in a given period of time. Okay. Once we capture individuals, we'll basically mark them So on each of the lizards, we'll probably paint something using color, or you basically leave a mark using a marker or a pen, right? And then you basically release them back. Then you give enough time for the population to intermix so that the like lizards are mixing and going around here and there. And the next time, and then the second time, what you do is you go again and you again capture individuals. And now you just check for marks. So let's say in this first case, you captured 50 individuals and you marked all of them and you release them back, right? So in the second case, let's say again you catch 50 individuals. Out of them, you see that 10 are colored, right? Colored or marked. So based on this, how do you calculate the number of individuals that are there in the population? So what you do is the number of in the second time when you are catching the individuals, the number of marked individuals or animals that you are catching divided by the total number of animals that you have caught in the sample. So for example, 10 divided by 50 here, because 10 individuals are marked out of the 50 that we captured in the second time, will be equal to total number of marked individuals in the population. So the first time we marked 50 individuals. So 50 divided by n, which is the population size. So from this equation, we can get that n is equal to 50 into 50 by 10. Right. So which is basically 250. So this is how my recapture method works. So you first, at time one, you basically capture individuals, you mark them, and you release them back. At some other point of time, time two, after some time, you basically capture individuals again and check for marks, and the ratio of the individuals that are marked and the individuals that you are catching uh, combined with the factor of how many individuals of the total population are marked in the first time will give you basically the total population size. Okay, so we have a question. In PPS, if greater unit is picked, then more time is required for study, leading to exposure of less variety. Uh, can you elaborate what you mean exactly, Rakshit? Take the last part, leading to exposure of less variety. What do you mean exactly? Less variety of species. So, I'm still, yes. So basically, all of these are the same species. So we are not looking at different species here. We are looking at a population. So we started off with population. So basically, we are looking at the same group and same species which are living in a geographical area and are interbreeding. So all of the things that we are calculating here are only for the same species. So I don't think the variety part comes into play unless like, yeah. So in PPS sampling, we are basically sampling one species, but based on the, let's say, age of the species, there might be differences in size. And we are trying to basically uh, use probability so that we are sampling more of these individuals and less of these individuals. Because there might be a huge number of saplings, but very few trees, right? And so, for example, in this area, let's say, we have some saplings, we have some basically medium trees, and we have some very large trees, right? There might be, let's say, 100 saplings. There might be 20 medium trees and 10 large trees, right? So when we are estimating the biomass of this whole region, we want all of these tree species to be caught because they will affect the biomass significantly. Whereas if out of these 100, if we, let's say, even if we miss out 40 or 50 of the small saplings, it won't matter to us that much in terms of biomass. So we are putting more probability of sampling these larger trees and less probability into sampling these smaller saplings. So all of them are of the same species. So there's no variety that is getting like missed. Right. Is that clear? Okay. Yeah. So as we were talking about the, yeah, thanks. Capture recapture method or mic recapture method. So this method assumes certain things. So for this relationship, for this equation to hold true, we have to assume certain things. So the first thing is marked and unmarked animals are captured randomly. So it should not be like you are capturing only marked individuals easily and unmarked individuals you are not able to capture easily, right? You should be able to capture marked and unmarked individuals equally. The second thing is mortality rate of marked and unmarked animals is the same. So you can basically uh, so let's say you mark 50 individuals and they have a higher mortality than other individuals. What might happen is more of these 50 individuals might get lost. So the next time you are capturing, you might not be capturing what is exactly the population size because more of these individuals are dying and less of the other ones are dying. Right. And the third thing is marks are not lost or overlooked. So you need to make sure that every time you are, uh, like the second time you're catching individuals, you are basically not overlooking any of the marks, or you're basically the marks that you're putting should not be something that will easily go away. Right. So, for example, like permanent markers and stuff generally stays for a while in many animals, whereas in some animals, these things might just go off. For example, like snakes or other animals, when they shed their skin, if you have a mark on their skin, then those marks might be lost. Right. So the third method to estimate the population size or population density is the removal method, which is Um, so first we'll look at the assumptions of the removal method. So the first thing is there is no immigration or emigration of animals. So the animals are not affected by animals coming from other places or going to other places. The death rate and birth rate are negligible during the entire part of the experiment that you are carrying out, the removal experiment. The trapped animals are permanently removed from the system because generally removal is like an invasive method of sampling where you basically remove animals. Right. Fourth thing is the trap can be occupied by only one animal at a time. There is no interference of animals of the wrong species, and the probability P that a specified free animal will be caught in a trapping period remains constant for all animals and all periods. So there should not be some individuals that you are preferential catching or some time periods in your sampling design where you are preferentially catching more individuals. Right. So basically, at any given point, if you are removing individuals, so the amount of individuals that you are exactly capturing during your removal experiments So basically what you do is basically you set a trap and an animal comes and falls into that trap. Then you basically remove that animal from the population and you set a fresh trap and you wait for another animal to come. Right. So this removal or the rate at which you are getting individuals will depend on the number of individuals that are already there in the population. So the rate of removal here, dn by dt, will be basically proportional to the number of individuals at any given point. So if you have, let's say, 100 individuals in a given area, then you are more likely to catch or remove more animals compared to if in the same area you have only 10 animals. Right. So then based on this equation, if we integrate it and we do this experiment, we can basically get, so T is basically the time here and not is the population number at the beginning and is the population number later. A is basically constant. So A and N naught can be estimated using regression removal experiments. It once you get the N and the T. So to measure, so measures that we talked about, the macro capture, the quadrant sampling, and the removal method give us the absolute population density, the absolute population number. If you want to get at the relative population density, we can basically use things such as traps. So for example, like light traps, right? We can use things like number of fecal pellets that are present in a given area. What is the frequency of vocalization of animals? Is the basically in a given area compared to another area. That means more animals are present in that area. Then pelt counts, then number of fishes that you're catching per unit of fishing effort. So for example, let's say you have 2 ponds where you want to see relatively if A and B where the number of fishes is more. So what you can do is here you can put a net and you catch, let's say, 20 fishes. Here you go, you put the same size of net and you catch, let's say, 50 fishes. So that means B has more number of fishes. That's why you are able to catch more fishes there. Right. So catch per unit of fishing effort. uh, Number of artifacts. Artifacts are basically things that or byproducts that these animals might be producing or using. So, for example, one way to estimate the relative density of bird population is to look at nests. How many nests are there in a given area? Right. Mm. Other thing can be questionnaires for people. So asking people how many of these animals do you see here regularly, and then asking the same thing in another area and comparing it to it. Then percentage of plant cover. So you take photos and you estimate the percentage of plant cover in that. Uh, Another thing can be frequency of quadrats where the species is found. So for example, let's say you sample 100 quadrats and in 50 quadrats you get uh, one species in a given area. In another area, you get, let's say, in 10 quadrats, you get the same species. So that means the population density is higher here and lower here, right? Feeding capacity also is an indirect measure of population density and can be used to measure relative population density. So you leave out, let's say, 10 kg of meat and you see how much of it is gone by a day or two, right? In an area that is, let's say, dominated by wolves or something, and you want to measure how many wolves are there. So basically, if more amount of bait is taken from a given area, that means more is the population density there. Right. So there are certain other things that we also look at for demographic information, such as age pyramid, which is basically the distribution of various age groups in the population. So what we have is like, let's say 5 to 10 year olds, how many of them are there? Then 10 to 20 year olds, how many of them are there? So the bar sizes represent what is the number, 20 to 30, 30 to 40, and so on. So what this tells us is basically less number of individuals are 30 to 40 years of age. The highest number of individuals have the ages between 10 and 20 and so on. Right. You can also get at the crude birth rate, which is basically the number of live births per 1,000 individuals, the crude death rate, which is the number of deaths per 1,000 individuals, the fertility rate, which is live births per 1,000 females of reproductive age, the age-specific fertility rate. So this is again the fertility rate for specific age classes in the reproductive age. Then the total fertility rate is the live births per female individual completing her reproductive age. If she followed the current age-specific fertility rate of the population, then we have something called replacement level fertility, which is basically the average number of offsprings that a female individual is must produce so that the population is completely replaced for the next generation. So let's say you have two individuals, male and female, and they are basically having two children. So in that case, so all the parents, so there are two people in this generation and two people in this generation. So the population is getting replaced in the next generation. So basically the replacement level fertility should be greater than equal to 2 so that we know that the population is expanding. If it's less than equal to 2, that means the population is in decline. If it's less than 2, the population is in decline. So let's say there are two individuals in the parent generation, but here only one. So the population is declining, right? Then juvenile mortality rate, which is basically number of deaths of juveniles per 1,000 live births. Life expectancy, which is basically the number of years that an average individual at in that particular population can be expected to live. Right. When we talk about life expectancy, there are two things, two factors. One is physiological longevity, the other one is ecological longevity. So physiological longevity is the average age or longevity of individuals of a population living under optimum conditions where individuals are dying of senescence. So there is no predation, no disease, nothing else. So individuals are living under optimum conditions and they're living out their life. So they're dying only after they're getting old. Okay. So this is known as physiological longevity. So the age that you calculate that way. Ecological longevity is basically the empirical or the observed uh, age that you get from individuals that belong to a population and are living under certain given conditions. Right. Uh, Other things that we might want to calculate are things like immigration, so the number of individuals coming in or going out, net migration, which is basically immigration minus immigration. If it is positive, that means more people are coming in than leaving. If it is negative, then more people are leaving. Natural increase is basically number of births minus number of deaths. Population growth, as talked before, is births plus immigration minus deaths minus immigration. So birth and immigration add to the population and death and immigration decrease the population. We also talk about population growth rate. So which is basically how much is the population growing every year or like at over a fixed period of time as a percentage of the population size. So generally it is expressed as like percentage per annum, percentage growth per annum. Right. So we need to measure all these things and to measure all these like population size, population density and all that, we need results or measurements that are both precise and accurate. So what do we mean by precise and accurate? So for example, let's say a gunman is shooting this target. Okay. Let's say there are four people, A, B, C, D. When A is shooting, what is happening? A has fired 5 shots. One comes here, one comes here, one comes here, one comes here, and one comes here, let's say. Then we have, so this is for A, this will B, we'll have C, and we'll have D. So let's say B is a good shooter. He shoots 5 of the bullets here. Let's say C shoots everything here, and D shoots everything all across. Right. So precision is basically cases such as B and C. B and C are precise because they're shooting at the same location again and again. Right. So when you're measuring things, if you're arriving at the same value or similar values again and again, the measurements are called precise. So when you're measuring, let's say, length of a table or something like that, and sorry, length of a pen or something like that, and let's say you measure it as 10, 9.8, 10.2, 10.3. And let's say your friend is measuring it again using some other ruler or some other method, and your friend is measuring it as, let's say, 10, 15, 5, 20. Right. So these results are more precise than these because these are close together. Right. Uh, B and C here are precise. A and D are not precise. Again, accuracy means the average of all the measurements that you take should come close to the like original value or the measured observed value, right? So in this case, B is accurate because B is shooting in the bullseye, and D will also be accurate because the even though it does not look like D has done a good job, the average of all these 5 points if you take will fall somewhere around here, right? So B and D are basically accurate. So in this case, let's say the um, length of the table is 10 centimeters. You are also more accurate than your friend because basically 30, 50. So if you do an average of this, it will come much closer to 10 than if you do an average of this. So this is more precise and more accurate compared to this. This is more. It's not precise, not accurate. Right. So the next thing we need to know is something called bias. So basically, for example, in the case of C here, C has very precise measurements, but it is not accurate. So the distance from the actual value to where exactly we are getting a value here is basically called as the bias. So for example, in the case of table, let's say a third friend came and measured the table and he measured 15.1, 15.4, 14.7, 15.8. So these are again very precise because they are again very close together, but they are not accurate since the and your friend is consistently getting values that are close to 50. So the difference between 15 minus 10 here, which is 5, is basically the bias of these measurements. Right, how far away it is from the accurate measurement. So to remove measurements, we need to calibrate our instrument or change our method of measurement. So your friend here can basically take a better label scale, right? Or the shooter here can basically the bias here that might be coming in for shooter C is basically their barrel is probably bent to the right. So what they can do is either straighten their barrel or choose another instrument or another gun. Right. So that's all for the first lecture. Does anyone have any questions till now? Can capture and recapture method be used for territorial animals? It can be used, but the thing is, you need to make sure that you are not doing your capture recapture in only one territory. So if you know that an animal is territorial and you have an idea of like how big a territory might be, right? Then in some cases, you can use mark recapture by increasing the size of the area that you are sampling. But if an organism has a very large area, territorial area, for example, tigers and stuff, they might have very large ranges and very large territories. It might be difficult to do mark recapture for tigers, so you try to do other things. That's also something we'll look at. Basically, some methods might work better for some species, and other methods might work better for other species, depending on how they are behavioral. So how is juvenile defined? Juvenile is generally producing. So generally, when you define animals like stages of life, uh, juvenile will be any individual of a population that has not yet attained sexual maturity, but it is kind of independent of its like parents. Not independent as such, but actually individuals that are basically not infants anymore, but have not yet reached sexual maturity are generally defined as juvenile. And it is species dependent kind of. So like, um, it's not exactly species dependent, but like some species might have uh, juvenile periods that are longer than other species. It's mostly to do with reproduction. So next we'll look at population growth and regulation. So here, sir talked about a few questions and how we can uh, work out a few things. So let's say how using these sampling data, how can we compute the density of animals? So here we have a question. A park manager conducts a population estimation exercise within a protected area. He samples 9 quadrats with line transits and obtains the following density estimates for sample. Okay. So we have like beat is basically a area that is covered by a forest guard. So beat is like an administrative unit of sampling. So it's defined by the forest department, and that is something that people can also use. And the forest department, I think, regularly uses. So we have all these different beats, and we are basically seeing how many samples are present per square kilometer in each of these beats. So that is the data we have. Now we have to compute the average density of sambar in the whole region. Right. To compute the average density of sample, what we need to do is density of sample is given as density in the beats. So you sum the density of summers in each of the beats, and then you basically divide it by the total number of beats. So in our case, it will be basically 8 plus 5 plus 6 plus and so on. So basically 8 plus 5 plus 6 plus dot 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 till plus 6 divided by, we have 9 beats. So the total here would come out to be around say 55 divided by 9, which will almost be equal to 6.1. So that is the average density of sambar in that area. Right. The area of all the 9 beats. So the next question that was discussed is the group sizes of cheetal in the core and buffer zones of Corbett Tiger Reserve were recorded during winter 2009. Estimate the mean group size, standard deviation, standard error, range, and coefficient of variation, and comment on the results obtained. So we have basically 10 observations here from each the core zone and the buffer zone. So when you have basically a protected is basically the innermost area, and a buffer zone is the area outside the core zone, but not exactly fully anthropogenically disturbed. From here, you might start to get a lot of disturbances. But here in this protected area, this place might have only a few disturbances, such as, let's say, a few small villages or a few number of houses here and there. Right. So this is defined as the buffer zone, and this is defined as the core zone, protected area. So first we'll calculate the mean group size. So these numbers are basically the group sizes in each of the what do you say samples that we are taking. So to get the mean group size for the core, what we need to do is we need to sum all these different observations that we have for the core group sizes, and we divide it by the number of samples that we have. Basically, we do 26 plus 24. Plus 25 divided by 10. So this will turn out to be somewhere around 250, which is 25.2. And similarly, for the buffer region, if we do this, we basically buffer group size divided by number of samples. We basically have 2611. So here we note that the mean group size of the buffer region is 20.7, whereas the mean group size of the core region is 25.2. What we do is basically, so the standard deviation is given by x minus mean whole square divided by n root over, where x is each individual observation, mu is the mean. We are taking the square, we are dividing it by n, which is the total number of observations or samples. We are taking the root. So ST for core, if we do the calculation, we basically have 26 minus 25.2 whole square plus 25 minus 25.2 whole square plus 25 minus 25.2 whole square. This is a number of samples, and we take a root over of this. So this comes out to be approximately 1.249. So these calculations are simple. I've just done it with first. I'm just writing it so that things get done quickly. If you want, you can stop here and you can do your own calculations. Right, just to get at how it's done. If you have any doubts or any questions, please. Here again, we subtract it from the mean, which in for the buffer case is 20.7. Then we divide it by 10, which is the total number of samples, and we take the root of it. So in this case, it turns out to be somewhere around 11.385. So first we'll do all the calculations, then we'll think about what is happening. So the standard error is basically standard error is given as sigma by root over of n, which is basically the number of samples, and sigma is basically the standard deviation. Right. So here for the core, we are calculating the standard error. we have 1.249 as a standard deviation, and this is basically 10. So if we do this, it comes up in 5. And for buffer, we do the standard error. It turns out to be 11,385 divided by root over of 10, which is equal to 3.6. Right. Range is basically the highest value minus the lowest value. Right. So for core, for buffer, so for the case of core, the highest value we have here is 27, and the lowest value that we have is 23. For buffer, the highest value is we do 27 minus 23 equal to 4, 40 minus equal to 37. Now we calculate the coefficient of variation. So the coefficient of variation is given by the formula sigma by mu into 100%. So in this case, we have 10 samples from the core, 10 samples from the buffer, so we don't have any issues. But when we are carrying out, doing these things in real life, we might not have the same number of samples. So in that case, to compare what does these standard deviation and mean, basically how they relate to each other in these different cases where, let's say we have for buffer, let's say we had 5 samples, and for core, let's say we had 20 samples. So comparing the between them, we would need to compare the coefficient of variation, which is given by sigma by mu into 100. So how much percentage of the variation currently, right? So for the core, we have basically sigma percent, which is basically 1.249 divided by 25.2 into 100%, should have 0.56. And similarly, for buffer, we have 11.385 divided by 20.7 into 100, which gives us somewhere around 54.998%. Okay, so now we'll go back. We have made the calculations. So we'll look at it one by one. So the mean group size, what does this tell us? So the mean group size in the core is 25.2. The mean group size in the buffer zone is 20.7. This, on an average, if you encounter a group in the buffer zone, it will have around 20 individuals. But if you encounter a group in the core, it will have 25 individuals. So this probably indicates that there might be more individuals or more individuals grouping in the core compared to the buffer zone. Right. Now, the standard deviation, what this tells us is within the samples that we have for the core and the buffer, how are the group sizes varying among these samples? So the standard error also gets at the same thing. So higher value means that there is more like difference in the number of in the size of the groups for different samples, and a lower value means that all the numbers of individuals in each of the samples is almost approximately the same, right? See that the core has a lower value compared to the uh, buffer, which has a very high value. So what does this tell us exactly? This basically means that if we have, so we have the core and we have the buffer. So if we put like two samples here, the values that we'll get from these two, the group size values, will be similar. But if we put two samples here in the buffer zone, the values that we get might be very far apart or very different. Right. And then the similar thing we get for standard error. This is higher in case of buffer, and thus basically the estimates that we get from different samples in buffer are more varying. Right. The range basically our data is in a sense. So a small range means that all the samples have comparable values of group sizes. A very high range means that the samples have very like high differences between the group sizes. Now the coefficient of variation again tells us like how much is the standard deviation compared to the mean. So how much like is the variation very high compared to the mean that we are getting? Or is the variation very low compared to the mean the value that we are getting? Right. So this also points towards like buffer having very irregular and very, what do you say? Uh, Spread out values of sample numbers. All of this is also visible that when we are doing these mean and standard deviation, standard error calculations, we are basically getting at a more quantitative level. We are seeing what is happening. So from these, what conclusions can we draw? So the first thing we can say as before is that the core and the buffer, the core is more homogeneous because there is not a lot of variation. The range is also very low. The the coefficient of variance is low, coefficient of variation is low, the range is low, the standard error, the standard deviation are very low. So what this basically means is that if you take samples from anywhere from the core, you will almost get similar values of group size. So the group size is homogeneous in the core. Whereas for buffer, since these values are opposite, if you take 4 samples from different places, right, you might get very different values in all 4 of these samples. Now, these differences might be arising because, as I was telling before, in buffer zones, you might have, let's say, villages or like houses. So these houses and villages will have a bigger zone where they're basically uh, impacting. So for example, like these core areas, they can still go to some of the areas of the buffer zone, let's say to collect, what do you say? So yes, there are 2 houses here. They go into this buffer zone to basically collect firewood or like leaves of plants for mulching and stuff like that. So basically the presence of these and humans in this area leads to a low group size in numbers here compared to a. So that's why we probably see these differences. So these are like some, and the other thing is here we have around 25.2, here we have 20.7, which is the group size. So basically, if there is not a lot of disturbance, the group size might be higher or uh, compared to if there is some disturbance, for example, in the buffer zone. Right. So these are the conclusions that we can draw. Right. Regarding the core and the buffer zones. Does anyone have any questions? So next, I'll discuss some basically methods that are utilized to sample herpetofauna in like, especially in rainforest. So we'll talk about all of them. Basically. So opportunistic encounter is basically you're randomly going on walks and you're seeing a species somewhere, right? So let's say you're taking a walk, you're just going around, you don't have any motive in your mind, you're just going around in a trail in the rainforest and you see certain species. That is an opportunistic encounter, right? Directed survey is basically when you go around looking for a specific organism or a specific animal, right? Mhm. Or you go around looking at a given area and seeing what animals are there. A is basically when you have like directed surveys and opportunistic encounters, you take the data together and what do you say? Try combining these and getting at how many exactly different species are there in the given area. So basically estimating the complete number of different species and their population characteristics will be a complete species inventory. Visual encounter surveys are basically where you're going around uh, and looking for animals. So basically, frogs or something, you might go into an area you might find out like certain rocks where basically, so sorry, for lizards, let's say you are going to look for lizards, you know that rocks. So what you do is in that given area for a constrained amount of time or something, let's say for 1 hour or something, you just go around, you flip rocks and you see if there's a lizard below the rock. Right. So that is visual encounter survey. Quadrant sampling we talked about in detail. You basically have a lot of different sampling units and you choose certain samples from that and you estimate basically the population size and density there and extrapolate it to the population. Distance sampling is like things like transects. So a transect is basically of land, you basically draw a line or a path and you basically walk on that path and walk on the straight path, and on your left and right, you keep looking out for species and animals that you're finding. Right. Uh, Patch sampling is or adaptive cluster sampling is basic technique where basically, let's say you have this, sorry, uh, so you have this whole sample a certain species. What you do is you go to a small area in this and you see if the or a patch here and you see if the species is present. If the species is present, let's say you find the species here, what you do, you look at all the patches that are neighboring this, all the neighboring patches. So there'll be 9 of them. And you see if the species is present here. So you see the species is not present here, 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 but it's present here. So next what you do, you look at all the neighboring patches of this patch. You find the species here and here. Then you again look at the neighboring patches. And let's say you don't find anything. So basically now this region where you found all the animals is basically the spread and you're having like individuals here. Like this is known as patch sampling. You choose a patch, you look for a species. If you find it, you look for adjoining patches if the same species is present. And you do this till you have, till you encounter a patch where there are no species in the adjoining area. Right. The next sampling method is called a audio strip transect. So basically, you go on a transect and you record audio using a recorder or something, and based on the like vocalization, you can identify what all species are there. And based on the number of vocalizations or the number of calls that you might get in the audio recording or when you are just going around collecting data, you might basically uh, estimate the abundance of the population also. The other thing was mark recapture method, as we talked about. You basically capture some animals, mark them, release them. Then afterwards, these animals are again basically captured, and you count how many of the individuals are marked and how many of the individuals are not marked. Right. Then you have something called pitfall trapping or funnel trapping, trapping with drift fences. So generally, what happens is on the ground, you basically dig holes and you put like dig pits or holes, and in these pits you basically put some let's say water and detergent or uh, some like formaldehyde or some solution where basically like insects coming might fall into the pit and die inside. So basically, based on how many insects are dying of how many different species, you can estimate what is the population density of different species of insects in that given area. Cover board surveys in this method, what you have is basically you have cardboards or cover boards, which you take into a given area and then you randomly just throw it around. And wherever those boards fall, in those places, you have to sample for a given animal. Right. The next thing is surveys at breeding sites. So basically for herpetofauna especially, ponds and stream habitats can have amphibians and reptiles. especially during the breeding season. So going to these kinds of places or visiting these kinds of places during breeding season helps you basically usefully estimate the number of species that might be present, because many of the species will aggregate around these places. The next method is quantitative sampling of amphibian larvae. So basically, you go to a pond and let's say you want to estimate the number of frogs that are present. You can estimate the number of tadpoles and basically use that as a proxy for the number of frogs. So you're basically sampling the number of tadpoles, and from that you are getting at the number of the adults that might be there in the population. The next method is basically using instrumentation such as camera traps or active and passive sensors, which can be deployed in different areas with or without baits. So basically, let's say you're camera trapping for a tiger. It might not be very easy to see the tiger. So what you do is you leave out some meat or you tie a goat near to the camera trap. So if the tiger comes and eats the goat, you get a photo. Yeah, but like for herpetofauna also you can do the same thing. Different frogs, you can give different food items or things that the frogs are attracted to, and basically insects and stuff that the frogs are attracted to in front of the camera trap, and basically wait for them to approve. Right. Now, as someone was asking before, also, uh, yeah, like can we use capture recapture for territorial animals and stuff like that? So basically, it is important to note that even though we have all these sampling methods, some of them might be more effective than others, depending on the species and the characteristics of the animals that you are trying to measure. Okay, so for example, if like there are areas which have a lot of water pools, and you want to sample the herpetofauna that is present there, market capture might be a very difficult task because there might be thousands of individuals, and basically, what do you say? Um, you might catch a lot of individuals at the same time, and they might not be moving around a lot or mixing with the population. Some of the assumptions of the market capture method might be violated. So what you can do here is basically you can put up a audio recorder, and you can basically record the audio and basically use that to identify the species or the number of individuals of a given species. On the other hand, if you have a species which is silent and there's only one breeding site where the species is found, you might just go there and count the number of individuals if there's a low number of individuals, and or you basically do a transect or something like that in that area without audio, basically because it's silent species and so on. So basically, based on the behavior, physiology, and the ecology of the organism that you are studying, you have to adapt your methods and choose better methods that can help you basically um, get at the population parameters. Right. Another important method of trapping insects is called pan traps, where you basically, like pitfalls only, you have these colorful boxes where you have soap water solution. So soap water solution basically reduces the surface tension of water so that insects can't land on it. And if they try to land on it, they go inside and basically drown. So pan traps are very effective for pollinators because pollinators are attracted to all these different colors. So pan traps can be used to catch pollinators, whereas pitfalls and funnel traps can be used to uh, catch all different kinds of insects that might be present. And based on the proportion of animal individuals that you get for pan traps and for insect traps, you can determine what is the proportion of pollinators among insects in a given area. It things like that. The other thing is because they are very low cost, it's just a paper cup. They are very reusable. You can just wash the cup and use it again. And they are very effective. As you can see, there are a lot of bees that have been caught in this trap. So these are very widely deployed and very important methods of studying insect populations. Um, So let's say we have some question like, how will you plan to monitor the population of a species which has distinct marks? So for example, a tiger might have, tigers might have different striped patterns. So based on the striped patterns, you might be able to identify each individual of a species like tiger, right? So in that case, it makes our math recapture um, protocol slightly easier because we don't need to capture individuals and mark them exactly. They are already marked for us since we can identify them individually. What we need to do is basically go to some place, take a few photos, then go again there some other time and take a few photos and compare if like we are getting the same individuals or we are getting different individuals. Right. So the question here is describe how you will plan population monitoring of a species with distinct marks. So since the population has distinct marks, which are probably visual different identification marks, so we can easily use camera traps. And as mentioned, we can use mark recapture method, but we need to keep in mind two things. The first thing is the ecology and behavior of the species. So is the species aggregating at certain places? So for example, peacocks aggregate in certain places for lecking. Right. Black bucks also aggregate for lecking. So if we are basically looking at species like that, which are aggregating or congregating at certain sites, we can put our camera traps there. Or if we know that a tiger follows a certain trail, we can put a camera trap there near the trail. Right. The second thing is we have to also be careful of the abundance of the species. If there are not a lot of different, if the species, if the abundance is not that high, if the number of individuals of the species are not that high, uh, we can actually, and we can identify individuals, we can actually count all the individuals that are present of that given species, which is something that has been done for tigers. Almost all the tigers in India have a name and they can be distinguished based on their stripes. So basically, these are the two things that we have to keep in mind, and we can calculate the exact numbers or the population trends by basically either calculating the number using matrix capture method as I was telling you. So you basically go to this place, you take let's say 50 photos on day one. Okay. Then you go to that place again. You take 50 photos, I think, 50 photos of different individuals. Right. You go there again in day, let's say day 20 or something, and you again take 50 photos. Now, what you see is of these 50 photos, how many photos have you seen earlier? So let's say 10 photos are of individuals that were also present before. So based on that, you can basically calculate what is the population density and population size. Another thing you can do is if you can see how many males, females, and juveniles are there, based on that, you can have like the age and sex pyramids you can build for the population, and you can see how the population is it increasing, is it decreasing. So basically, generally, if the age pyramid is something like this, where there are a lot of species, a lot of individuals that have a lower age compared to a lot of very few individuals having a higher age. This means the population is increasing. But if the age pyramid is something like this, where there are very low number of individuals in the lower age groups and very high number of individuals in the higher age groups, that means the population is in decline because most of these animals can't reproduce and will die off. And these animals are not enough to replace the animals that are there in the other age groups, right? Whereas here, even if these animals die, there's no concern. These animals will, like these young people, animals will basically reproduce and basically lead to more number of species. And we can also have age pyramids which are like this, more or less rectangular, where there's not a lot of variation or like there might be some variation like this or like this, but this age, so basically the adults of the population and the juveniles are basically almost the same. So the population is in a constant mm, number. It's not increasing or decreasing. Right. So these all things we can see to monitor the population. Right. So next we talk about, so these are of population demographic characteristics and stuff. But how do we model growth of a population? So this is basically the equation that is used to, this is basically exponential equation that is used to basically model the growth of a population. So here NT is basically size of the population at generation T. NT plus 1 is size of the population at the next generation T plus 1. R naught is the net reproductive rate, which is basically the number of female offspring produced per female per generation. So what is happening is if R naught is 1, that means the population is constant. So the population is not increasing or decreasing. If R naught is less than 1, that means if you multiply NT into something less than 1, it will decrease, right? So the population is decreasing generation by generation. If R naught is greater than 1, then the population is increasing. So generally, R naught values initially if the population is starting tend to be greater than 1. So in that case, what we see is the population will tend to grow. And it will be some sort of exponential growth because this n will keep compounding over time. So let's say here you have 10 individuals, and let's say your R is 2. So in the next time step, you will have 20 individuals. But in the, so this is t equal to 0, t equal to 1. But in t equal to 2, you have 40 individuals now because your R is 2 again, and 20 into 2 is 40. So here in one time step or in one generation, you have an increase of 10 individuals, whereas here you have an increase of 20. And this increase keeps on increasing, and thus you have an exponential curve here. But actually in populations, in like real life populations of animals, what happens is this is not true. So the R value keeps on changing. So basically a population cannot keep on increasing like this exponentially because the resources are limited. Right. So what happens is people have proposed this logistic growth equation, which basically models populations much better. How this looks is something like this. It's a sigmoidal curve, and this value is basically called the K. The asymptote is basically called the K. So initially you have a lag phase where basically the population is not increasing that much. Then you have a log phase where basically the population is increasing exponentially because so the initial phase not a lot of increase because there's not a lot of individuals. When there's enough individuals, after some tipping or critical point, there's a log phase where there's exponential growth. And then as the resources start to become more scarce and scarce, the population growth tends to slow. And then it slowly basically asymptotes around this value called K, which is called the carrying capacity of the environment. So basically, how many individuals of that species can the environment support at that given point of time? So we have this equation of logistic growth, which models population growth much better than the previous equation. Right. And here is the population size at time t, k is the carrying capacity, and r is the resource requirement. Okay. So yeah, these are the equations that are used to basically model populations, such as the Lotka-Volterra equations, which are used to model how prey and predator populations functions in increase or decrease, the dynamics of prey and predator populations. So here, basically, V is the prey population at time t, P is the predator population at time p, alpha, beta, R, and Q are constant. So according to Lotka-Volterra equation, is positively influenced by the prey population. and negatively influenced by the predator population. So alpha into. So if there are more prey, more will be caught. If there are less prey, less will be caught. If there are more predators, more will be caught and eaten. If there are less predators, less will be caught and eaten. So that's why there's a V and there's a P. Now for predators, since predators are using prey for food and competing with other predators, beta VP is positively correlated to the change in predator numbers. So if there are more predators, sorry, if there is more prey, that means there will be more predators because there's enough food. If there are more predators, that means they can reproduce, producing leading rates to more predators. And this is inversely affected by the uh, number of predators because they are again competing with each other for food. This is minus Q into P. So this is the Lotka-Volterra equation. Basically, it produces a graph like something like this. So for example, let's say we have cheetal here and we have tiger here. So basically this y-axis is number of individuals and the x-axis we have time. So if the cheetal population is increasing, after that, after following a slow lag, the tiger population is also increased. But since the tiger population is increasing, the cheetal population decreases, right? Since the tiger population is increasing, the cheetal population will decrease because there's a minus factor here. So this decreases. Then after a point, since there are not enough cheetahs, the tiger population also decreases, which leads rise to another cycle of increase in cheetal population and so on. Okay. So these Lotka-Volterra equations model these predator-prey relationships. So that was end of the second lecture. In the third lecture, we look at population studies and we look at a case study. So does anyone have any questions till now? So population studies can be of two types. So if a population of animals, number of individuals is changing quite a lot, then we have problems of dynamics where we want to know how does the population change with time? Why is the population changing with time? Next, if we have a population that is not changing that much, then we can ask problems of statics, which basically is like asking things like what determines the equilibrium conditions and what is the average value of the population and things like that. Right. So examples of problems of dynamics might be like, so basically, what is things that we might ask in a dynamic population are like, do the changes occur in a particular time of the year? Do the changes occur at a particular life cycle of the organism? The agents that are operating at these times and stages, right? So, for example, let's say we are looking at cheetahs and we are seeing their population increasing and decreasing. So we first ask like, does the change occur in a particular time of the year? A better example might say uh, pollinator, right? The population density or the population size of pollinators, right? So the first question you ask is, does the change happen in a particular time of the year? So do you see more number of pollinators at a particular time of the year and less number of pollinators at other times of the year? That might be true. You might be seeing more number of pollinators around the times where plants are flowering. If they are flowering at the around summer months or something, if more flowers are flowering, then you might see an increase in the pollinator species, right? For example, in butterflies and stuff. The second thing is, does the change occur in a particular stage in the life cycle of the organism? So for bees, I don't think that matters quite a lot. Like, let's say we take bees as pollinators. Uh, I don't think the increase in population matters that much, but for uh, some other organisms that might matter. So different life stages of organisms like larvae and cocoon and all that might be affected differently by different things. And therefore, there might be some changes. For example, if there's an increase in, uh, for example, let's say we are talking about something. Let's say the butterflies are predated upon by birds, right? But these birds don't eat the cocoons of the butterfly, right? In the population of butterflies due to birds, that will happen only after they have become adults. It will not happen before that, right? So these are the kinds of questions that we can ask and clarify things, right? The third thing is what are the agents that operate at these times and stages? So for the pollinators, might be flowers, might be like other birds that are preying on them and stuff like that. And these things might be affecting how these pollinator numbers vary across time, right? So agents can be either extrinsic or intrinsic. Extrinsic agents include like conditions such as weather, is it favorable, is it not, predators, parasites, diseases, the amount of food or resource that is available, if the uh, for example, if there's extreme weather conditions like rain and all, if the animal is not able to like find shelter, die off due to hypothermia or things like that. Right? Now, intrinsic agents might be things that are behaviorally happening or physiologically or inside the animal that are happening. Right? Uh, yeah, metabolic changes or changes that might be happening inside the organism also. Now, when we are looking at static population, the population size is not changing, so we don't need to look at dynamic factors, but we can experimentally manipulate the habitat to look for why the population is static at this number. So, for example, let's say we have an elephant population in some area, and let's say there are only 40 individuals. And we see time and again, let's say we record it 20 till let's say 2024, we have been monitoring this area, and every year we see that the population of elephants is around 40. It does not change. So now we want to know why is it around 40? Why is it not more or less? Why is it not increasing? Right? Because there might be enough resources, but we don't see an increase. There might be another area of the same size where there might be 160 elephants nearby, right? Which might have similar conditions, but this area does not have that. Here only 40 individuals are present year after year. So we want to know why. So what we do, let's say we get all the elephants out and we basically test if they have, what do you say, some parasites or other things on their body. They have two kinds of parasites, right? And let's say they are A and B. So what we do is we take an elephant and we basically remove parasite A. We give it medicine to remove parasite A, and we basically see like how it is affecting. So we take a few elephants, we remove parasite A. We see that if the population number is increasing or decreasing, or if basically the animals are dying or not. So basically, one reason why this population is stuck at 40 might be because juvenile elephants are dying parasite. So what we'll do is we'll take juvenile elephants, two or more elephants we'll take out parasite A from them, and we'll see what is happening. Are the animals dying or are they staying? Let's say they continue dying. So we know that A is not affecting. But let's say we take few other elephants and we remove parasite B. And now we see that the animals are surviving juvenile stage and reaching adulthood. So that means this B parasite is affecting the population of elephants. And that is why this is constantly stagnant at a rate of 40, at the number of 40 and not increasing because the juveniles are dying off because of this parasite. Right. So these are the kinds of studies and the kinds of things that we can look at for static populations. We can manipulate the habitat variables, look at like resources, predators, parasites, and are affecting the population to see if it's why it is getting staying there. Why is it constant? So, so I talked about the locust case study. I'll just briefly go over it since we are almost out of time. Yeah. So locusts are generally certain species of stage. Locusts have two phases. One is a solitary phase where they are basically colored like this. They are low numbers and they are not threats to agriculture. Earlier, people thought named it Locust danica, and they also have a gregarious phase where they're basically found in large swarms. They are nomadic and they are pests to agricultural land. They just go around from one area to another area, devastating crops. So earlier, this was thought to be a separate species known as Locusta migratory. Okay, so uh, people assume that basically these solitary animals are locusts, and basically these animals are other kinds of locusts that are basically. So these solitary animals are basically grasshoppers, and these are basically locusts, which are the agricultural pests. But what happened was uh, earlier when people started studying these animals, so the first thing you need to do in a population study is basically describe the population, right? Describe the different characteristics and things like that. Sorry. Plutarch was one of the first people who worked on this. He basically tried to detail about the population of locusts and grasshoppers that are found. Okay, so he basically looked at larvae and he said that mm, larvae are of three types, three coloration: migratoid, green, and danicoid. Sorry. Mm, Basically, then he goes on to describe what are the characteristics of uh, migratoid larvae, what are the characteristics of like greenish larvae and danicoid larvae, how do they look and all that. Um, so basically, this was one of the first studies of the two forms. Then in the same study, Protognev says that what he did was he basically took um, a lot of these larvae from different migratoid grasshoppers or locusts. Right. And then he put them in different cases. One where there was just one solitary larvae, one there were where there were like many larvae. So, for example, the larvae were kept from the first stage in small cases at a density of 30 to 50 larvae to 40 to 460 to 600 cubic centimeter of space. A typical migratory coloration is invariably obtained. In experiments in which the density of the population is less, so he also had experiments where there was some medium density. Okay, let's say 20 larvae to 2000 cubic centimeter. Okay, in this case, you observe that the larvae are migratoids, are of migratoid coloration. Okay, in this case, you observe that there was migratoid larvae, there was some green larvae, and there was some danicoid larvae. So this is one of the initial studies. So people are still thinking that these two in are basically different species of animals. They are not the same species. So in this study, what Plutarch showed was that if you take larvae from any of these, from either these danicoid or migratoid variants, right? Mm, and you grow them in different density of larvae, the coloration changes. So if there is more crowding of larvae, then you get migratoid coloration. If there is less, one solitary larva, it will basically become green. And if there is less amount of crowding, like some density but not very high, then you get a mix of all three coloration of larvae. Right, you get migratoid, you get green, and you tanicoid, and you get something in between also. Right. So what this basically tells us is that probably these two species are the same. So for the first time, he was able to say that probably these two species are the same, and that is why it is giving rise to, like, eggs of this species is giving rise to um, larvae of another species, like assumed species, under different conditions. Right. Then this guy basically, VP Uvero, what he said, he basically was the first one to characterize all these states. So basically, he found three unstable biological states of a locust. The first is phasis solitaria, which is the solitary phase, which is the extreme form by which the species is represented in a given locality when only isolated individuals are present and no swarms exist or have existed in the at least in one preceding generation. Okay. Then there's something called a phases transience or the transient phase to be applied to the form occurring in the given locality when the species is on an increase and its individuals are beginning to form loose aggregations, or on the decrease and the swarms are becoming loose and tend to scatter either in the hopper stage or shortly becoming adult. So basically, transient phase is basically when it's transforming from this solitary to this gregarious or like swarming species, swarming groups, basically. And the third phase is basically phases gregaria, the gregarious phase, where basically there's a large amount of swarms and there's a very dense amount of these individuals. So what happens is the animal is solitary, then it goes into the transient phase. So this phase is called congregants when they are congregating. And this leads to the gregarious phase where they have large swarms and they're moving out. Then slowly these swarms are dispersing. So this again goes into a transient phase. Now this is called a dissociance phase because they are dissociating. And basically it again leads back to the solitary phase of the grasshopper or the locust. Right. But it's not very simple. So even though he was able to characterize all these phases and say that they are cyclic, there are a lot of things, lot of more complex things that are happening behind. So there are behavioral changes that are driving physiological changes in the larvae that are driving the color changes, like the migratory color or the green color or the danicoid color, and which finally result in the morphological changes. And sometimes you might have individuals that look like solitary grasshoppers or locusts, but they have gregarious behaviors. So they are swarming with each other. But we might also have gregarious looking locusts that are not swarming, right? So basically, starting from this case of when someone started describing the species, looking at their larvae, then another person looking at their different phases of the larvae and things like that, they were able to basically reconcile that these are basically the same species. And now people, uh, and then later people basically tried to figure out how these swarms of locusts are getting formed. So basically, um, sometimes due to random chance, they are mutually habitating. And because of that, they are coming close to each other, the laying eggs close to each other. So there's some larval crowding. And because of that, it leads to some activation in the like behavioral and physiological activation, which basically increases their propensity like to be excitable. It also increases the fecundity because there are more individuals that are now coming together to lay eggs and more eggs being laid. That leads to multiplication. It's a positive feedback loop which keeps on going. And after a while, what happens? We see observe these color changes, we observe other morphological changes. So the solitary population is now becoming gregarious. And after that, there are a lot of swarms forming. And because resources might be getting depleted by now, these swarms are emigrating to other different areas and then multiplying there. Right. So now people are basically looking at epigenetics and other behavioral analysis people are doing to basically see how these phase transitions occur in these individuals. So basically starting from the population, describing the different individuals, then describing their larvae, describing the phases of their life, to getting at how these phase changes occur, how the population changes as a whole, describing this vicious circle of formation of swarms. And now working on basically how these swarms are formed, like epigenetically or um, using like molecular markers or like behavioral changes. So this is exactly what people are doing. So this is like a case study of how things like population dynamics can be studied. So here basically people are studying how locust population changed from the solitary to gregarious forms and how basically these studies have been carried out since the past few years. Right. So that is the end of it. Does anyone have any questions? It was quite a lot this time, I think. A lot of different things. Okay. Does anyone have any questions? Okay. So we'll stop here. We'll meet again in the next class. Thank you for coming. Uh, yeah, we'll meet again next week same time. Thanks. Bye.